Excuse me, doctor. You are muted. We can't hear you. Excuse me, doctor.
Doctor,
Can you
I
hear me?
think you are muted.
We can't hear you. You're muted.
Okay, can you hear me now?
Yes, doctor.
Yes.
Thank you.
Yes.
Okay, so in addition, well, so recall where I talked about the
Actually, doctor,
Yep.
we couldn't hear you from the beginning, from the first couple of slides. We heard nothing, even from the learning objectives. So could you please go over that?
Yeah.
Thank you.
So the objectives of this lecture is to talk about the difference between T-dependent and T-independent antibody production by B cells. So this refers to the fact that some B cells behave like innate immunity. In other words, we are immediately born with antibodies even before we are exposed to antigen. And these are produced by these so-called B1 lymphocytes. They don't require T cell help. The T-dependent antigens are those antibodies produced by B cells as you would get after receiving a vaccine. With the help from the T cells, the T helper cells, in the form of cytokines, they will induce those B cells to undergo isotype switching from IgM into IgG, IgA, whatever, depending on the type of antigen. In addition to isotype switching, the help from the T cells will also induce further differentiation of those B cells to undergo mutations in the CDR antigen recognition portion of the antibodies to create high affinity antibodies. Again, this is why vaccines are so valuable in protecting us against bacteria and viruses. And these mutations are brought about by an enzyme called AID, or activation-induced deaminase, which again is activated through help from T helper cells. So this is why T help is so important in generating high quality antibodies in the T-dependent reaction. After these B cells differentiate into these antibody factories, which we call plasma cells, these antibodies will then bind to the antigen to remove them, and most of those plasma cells will die, but another portion will remain as long-lived memory cells, which will remain in our bodies for months or even till we die. Again, this is why vaccines are so valuable. And before we start talking about this T B cell help, you need to recall three. Oh, stop arguing, just listen. Right. One is that B cells not only make antibodies, but also that they are antigen presenters as well. So in other words, they can capture antigen and then take them into the cell just like a macrophage or a dendritic cell, and then process them and present them on class 2
Ahmed's little brother, can you please mute?
Yeah, let's mute. All right. For presentation to T helper cells. The other point to remember is that if you recall on the lecture about T helper cells, we had four major subsets: Th1, 2, and 17, and the regulatory T cells. I never talked about the TFH, or the follicular T helper cells. These are the subject of today's lecture, but bear in mind that these follicular T helper cells, TFH, are derivatives of Th1, 2, and 17. This means that within these secondary lymphoid tissue, remember that your Th cells that come out of the thymus, depending on the antigen that they see from the antigen presenting cell and differentiate into Th1, 2, and 17s, this process also happens within the lymph nodes and the spleen in relation to the B cells. So we get TFH1, TFH2, TFH17 type cells, although we don't call them those. We just give them this general name of TFH. But these different types of TFH cells will vary depending on the type of cytokines they produce. Interferon gamma for TFH1, IL-4, 5, and 13 for TFH2, and IL-17 for TFH17. These cytokines will determine what kind of antibodies the B cells make. So as an overview of this process, just like T cells emerging from the thymus come out as naive T cells where they haven't yet seen an antigen, but they bear a mature T cell receptor, B cells emerge from the bone marrow with a functional B cell receptor, which remember is IgM and IgD, right? So they have a potential to bind to antigen, but they haven't yet seen antigen. And just like T cells, they will circulate through the body under the drive of chemokines going from one lymph node to another looking for antigen, and also going through the spleen. But remember that these B cells recognize whole antigen. They don't recognize antigen presented on class MHC that was processed by dendritic cells or macrophages, right? But nevertheless, they still need to recognize antigen, whether they're bacterial cells or viruses, presented by an antigen presenting cell, i.e. a macrophage or a dendritic cell. The only difference is that in this context, these antigen presenting cells, the macrophage dendritic cell, do not take in the antigen and process them into peptides for the B cell. Instead, it just attaches to these antigens extracellularly on its surface, and this is recognized by the B cell receptors. Now, in order to understand the process of activation as a result of antigen recognition of the B cell from a naive B cell into a into an effective B cell, we need to look at the anatomy of one of these secondary lymphoid organs, like a lymph node. And you'll see that within the cortical region, in other words, the outside region, you have these things which look like boiled eggs. These are the germinal center, these are the B cell follicular areas. So this is where proliferation of naive into effective B cells occurs. The yolks of these B cell follicles are what are called germinal centers. Within these germinal centers is where further differentiation of these effective B cells into making high affinity antibodies occurs. Within the medullary region is where T cells reside, and this is where the T cells receive antigen presentation from dendritic cells and macrophages. So this is where the differentiation of the TH0s into TH1, 2, and 17 occurs. So these are called the T cell zones. So as I said, the naive B cell, which at this stage only expresses the IgM, IgD antibody receptor, sees antigen presented by macrophages and dendritic cells. These are specialized cells within these lymph nodes. So within the lymph node, we have these what we call follicular dendritic cells. They differ from the dendritic cells for T cells in that they're non-phagocytic and they can attach to opsonized antigens, whether these opsonized antigens are bound by antibody or complement components like C3b or C4b, because these follicular dendritic cells have receptors for the Fc region of antibodies as well as for these complement components. So basically, if you have, for instance, a bacterial cell that gets tagged by the antibodies or complement within the blood or within tissue, they will get picked up by these follicular dendritic cells within the lymph nodes and then presented or seen by the B cells through the B cell receptor, which will induce them to make antibodies. Now, let me see. Now, this delineation between T-dependent and T-independent antibodies is quite an important concept. And the initial T-independent B cells, as I said, come into play as soon as we are born. And they usually develop within the fetal liver. They produce predominantly IgM antibodies, and they produce throughout our lifespan. They are directed more against, just like PAMPs, they are directed more against sugar, long-chain sugars or fatty acids, not so much against proteins. And within the lymph nodes, we have a variant of these B1 cells, which are present in this area here called the marginal zone, outside of these egg yolk type structures. So this is where T-independent antibody production occurs. Now, with regard to the T-dependent interactions, where you need the activation of T helper cells, as well as the B cells recognizing the same antigen, this initial step occurs within what is called the marginal zone. This interface or this boundary between the T cell and the B cells. The T cells are receiving antigen exposure by the dendritic cells. Remember, the dendritic cells are in the peripheral tissues, they're capturing antigen, they migrate to these lymph nodes. In the process, they've phagocytosed the antigen, degraded them into peptides, and presented them on class 2 MHC in the lymph node. So in this lymph node, these dendritic cells that have captured this antigen, presenting the antigen on class 2 MHC to the naive CD4 cell. And this activates the CD4 cell into the Th1, 2, or 17 phenotype. Meanwhile, the B cell within the same lymph node is seeing the same antigen, and let's say it's a protein or a bacterial cell, but it might be recognizing a different epitope on that antigen, right? But nevertheless, well, that initial step will induce that B cell to make antibodies against that epitope that it recognized. But in addition, that B cell will also take in that whole antigen and process it just like that dendritic cell did, and maybe present the same epitope as that dendritic cell did for the T cell. So it's presenting now the same epitope on class 2 MHC, even though it makes antibodies against a different epitope on that same large protein molecule, okay? Now, through the action of chemokines, it will cause this B cell to migrate into this marginal zone. And meanwhile, that activated T cell migrates to the same region. This, again, under the action of chemokines. And this drawing together of the T cell and the B cell allows for that T cell to see the same epitope that it saw on the dendritic cell being presented by this B cell. So in other words, at this stage, the B cell is performing the same function as that dendritic cell did in presenting the same epitope. As a result of this interaction, the T cell gets activated, provides signals to the B cell to proliferate into these plasma cells. These plasma cells are are relatively short-lived, but they undergo this massive proliferation. So they increase in numbers and they actively produce antibody against that antigen. Okay. But again, this is more in a primary response because that antibody is predominantly IgM. This is why you always get IgM being produced in a primary response, the first antibody production. But as a result of this interaction, where the cytokines and the co-stimulation between the T helper cell and this B cell, the T cell then undergoes a differentiation, a further differentiation into the so-called follicular T helper cell. The B cell, again, through this interaction, also undergoes a change from the plasma cell into this more differentiated cell where it migrates back into the B cell follicle, into that egg yolk structure called the germinal center. So this interaction is dependent on two co-stimulatory molecules. One is called CD40 ligand, which I talked about already previously with the T helper cells. So the CD40 ligand is important as a cross communication between the T helper cell and the antigen presenting cell, predominantly macrophages and B cells. In other words, through CD40 ligand, the antigen presenting cell not only activates the T helper cell, but the T helper cell also activates the antigen presenting cell. In the B cell context, why we know this is important is that certain individuals are born with a mutation where they can't express a functional CD40 ligand. And what we see in these patients is that they can only make IgM antibody. So this reflects the importance of this ligand receptor to induce isotype switching from IgM into the other types of antibodies. Another important ligand receptor interaction is this molecule called ICOS, or inducible co-stimulatory molecule, where ICOS is expressed on the TFH cell and ICOS ligand is expressed on the B cell. This also delivers another set of signals to induce further differentiation of that effector T cell into a fully functional TFH cell, as well as into inducing the different changes within the B cell. So once both the TFH cell has developed within the germinal center, as well as the B cell migrating in this germinal center, what happens is that, first of all, that B cell can undergo isotype switching from IgM into IgG, IgA, IgE, depending on the antigen. Remember where in the first lecture I talked about B cells, that even though you get this VDJ rearrangement within the FAB region of the antibody molecule, all of the initial antibodies are IgM because the gene coding for the IgM-Fc region is the closest to the VDJ sequence, right? So that C-mu constant region is the one that is associated with this whole messenger RNA, and all of these others, the C alpha, C gamma, and so on, are cleaved off. But within these germinal centers, through the in the activation of this enzyme, activation-induced deaminase, what this enzyme does is it loops out this bit of RNA to remove the IgM and select C alpha, C gamma, or C eta to create a different messenger RNA to code for a particular antibody. But the important thing to remember is that this antibody has the same antigen specificity as that original IgM. The only difference is the Fc region has now got IgA or IgG or IgE attached to it, right? But the antigen specificity is the same. And this process occurs through this enzyme activation-induced deaminase, which is activated as a result of the T helper cell. So that is one effect or one important role of the T helper cell or the follicular T helper cell in providing help to the antibody producing B cell with regard to this isotype switching. The other important function the T helper cell plays is to induce this T cell, this B cell, to undergo what's called affinity maturation. Now recall that in the first B cell lecture, I said that once VDJ rearrangement occurs within the antibody receptor on the B cell in the bone marrow, those B cells are then exposed to self-antigen. And any B cell that binds to a self-antigen too strongly or too weakly will die through this process of clonal selection, right? Clonal deletion to remove any potentially autoreactive B cell. Now, the same thing happens with affinity maturation, but here we have the opposite effect now, in that only those B cells which bind to antigen, and in this case, this is this foreign antigen, only those B cells that bind with the strongest affinity will outgrow the ones with weaker affinity antibodies. And again, this is brought about by this enzyme activation-induced deaminase. What this enzyme does, not only does it cause cleavage or selection of the appropriate Fc region with regard to isotype switching, but it will also induce point mutations within the CDR antigen binding region of the antibody. So basically, through this enzyme, it's causing different affinities on the antibody receptor of B cells for this non-self antigen. Those B cells that produce, as a result of these mutations, a higher binding or affinity for the antigen will deliver a stronger signal into those B cells to cause them to proliferate much more extensively than the other B cells, which also specific for that antigen, but not with such high specificity. And in this way, the process ensures that only those B cells that deliver high quality antibodies for antigen will outgrow over the rest. And again, this is why vaccines are so effective in creating this high quality, high affinity antibody. This process is called somatic hypermutation, which refers to these point mutations as a result of this enzyme AID to create variations in affinity of the antigen binding region. So once these plasma cells, which are dedicated antibody producing B cells, produce these high affinity antibodies, they will then do their thing of capturing, blocking, or causing killing of these bacterial cells. But then you also get a subset of these B cells, which then differentiate into these so-called memory cells. And these memory cells can live within the peripheral lymphoid tissue or other types, or the bone marrow or other parts of the body without needing antigen exposure. They are long-lived cells. And as soon as they see antigen, that same antigen again, they will then undergo immediate full-blown antibody production and proliferation without the need of having T cell help and co-stimulation, etc., which takes a lot more time to generate. So again, this is why vaccines are so important. One thing which I need to just expand on is that this process whereby the T cell, which was induced by a particular epitope presented by the dendritic cell, is then presented with the same epitope by a B cell, even though this B cell recognized a different epitope on the same antigen. This process is called linked recognition, referring to the association of the T and the B cell to come together within this marginal zone. But the fact that the B cell is presenting the same epitope to the T cell, even though it recognizes a different epitope on the antigen, is similar to the hapten carrier effect. So remember, what is a hapten? If you ask what is a hapten, then you need to consider what does it mean to be antigenic versus immunogenic. So a hapten is antigenic, but it's not necessarily immunogenic. So it's antigenic in that it can be bound or recognized by an antibody, but it's not immunogenic, meaning that it does not necessarily induce an antibody immune response, right? So the hapten is generally a small molecule because this is unable to cross-link the B cell receptor. In order for a hapten to be immunogenic, it needs to be linked onto a larger molecule, generally a protein, which is called a carrier protein. And the reason why it makes that hapten now immunogenic is because a dendritic cell can capture that whole hapten carrier complex and present epitope to a T helper cell. The T helper cell can then provide help to the antibody producing B cell to make an antibody against that hapten. I'll expand more about this in another lecture where I talk about conjugate vaccines. Have you heard about conjugate vaccines in your microbiology lectures? So this is the basis of this conjugate vaccine, because with the conjugate vaccine against, I think, streptococcus, which is antibody against the polysaccharide on the bacterial cell surface, polysaccharides are generally antigenic but not very good immunogenic molecules. So to make them immunogenic, what they do is they tag them onto protein carriers. So this is the same principle. So in summary, the induction of isotype switching, whether a B cell makes IgG, IgA, or IgE, is dependent on the type of antigen, whether it's a bacterial cell, whether it's a virus, and also the type, or whether it's an allergen, like a pollen grain or a food particle, as in the case of type 1 hypersensitivity. This antigen dependency starts off initially with the antigen presenting cell. What the dendritic cell picked up, whether it was a pollen grain or whether it's a bacteria or a virus, that will deliver a particular signal to the T helper cell to become a Th1, 2, or a 17. Depending on the cytokines that those subsets produce, it then delivers to the B cell to induce it to make the particular type of antibody. IgE if it's a pollen grain or a food particle, IgG if it's a bacterial cell, IgA if it's within the mucosal tissue. And this isotype switching and the generation of these high affinity antibodies occurs within those egg yolk type structures within those B cell follicles, the germinal centers. All right? And the generation of these high affinity antibodies occurs through this process of what's called affinity maturation or clonal selection, because only those B cells which express the highest affinity for the antigen delivers a growth signal to those B cells to outgrow all of the other B cells, which might be specific for the same antigen, but not with as much affinity, so they don't get as much growth signal. And then the other thing to note is that separate from these B cells which need this T help, you have these so-called B1 cells which can make natural antibodies just non-specifically against particular antigens. And this is my opinion, but I suspect that in this COVID-19 crisis, these natural antibodies are what is the main protective antibody against this virus. This is why I don't hold out much hope for this vaccine, but still, that's just my opinion. So again, this is another way of looking at this B cell differentiation pathway, where you start off with this naive B cell which emerges from the bone marrow. Once it enters the lymph node and sees the respective antigen, it becomes a short-lived plasmablast, which occurs within those marginal zones, producing loads of mainly IgM antibody. But these plasmablasts are short-lived. But following signals and help from T helper cells, they can then differentiate into these plasma cells, producing high affinity antibodies, as well as differentiating into these long-lived memory cells, where when they see antigen again, they will jumpstart and quickly produce the same high affinity antibodies. This just refers to the difference within that extrafollicular marginal zone, this part compared to what's happening in the germinal center, where this part is where you're getting the short-lived plasmablasts producing the IgM, but within the germinal centers is where you're getting the further maturation into these the isotype switching on the high affinity antibodies. And this is just something about these follicular T helper cells. As I said, they are essentially lymph node or spleen variants of these Th1, Th2, Th17 cells that I talked about. They are they are specific to these secondary lymphoid organs. They don't migrate through the lymphatics or the blood, just like the Th1, Th2, Th effector cells. So they are only dedicated for B cell help. And they express these two important receptor ligand interactions which you should remember, which is CD40 ligand and ICOS ligand. So the CD40 refers to the cross communication between the T helper cell and the B cell, but also with macrophages. And the ICOS is predominantly between the T and B cells. That's it. It's a pretty complicated lecture, but if you have any questions, feel free to email me. Okay?